Okay, so this should follow along with your note packets that you have. If you have questions as we're going through, throw your hand up and ask. You're probably not the only one that has the question. Okay, you're just the only one with the balls to ask. All right, so the first thing that comes up here, of course this is, oh there it is. Are there makeup quizzes allowed on quizzes? So if you miss a quiz, if you're late for a quiz, do you get to make it up? No, good. So you guys paid attention, at least caught that yesterday or 2 days ago. Come on. All right, so Unit 1. Quick overview of what we're going to look at this unit. We're going to do a little bit of an introduction. We're going to talk about cells. You guys have probably learned about cells in high school. It's kind of what high school biology is all about. So we'll touch on a little bit of that stuff and kind of work our way into tissues. The study of tissues is called histology. Okay? Ology is always the study of something. Then we'll move into intracellular junctions, which are how cells are joined together. We'll talk a little bit about glands because they're made up of epithelial tissue, and then we'll look at membranes. Then we'll go into tissues, how they grow, how they die, in between how they repair themselves, that type of thing. And then lastly, this unit is basically the integumentary system. Okay, I won't lie to you, it's kind of a boring unit. Okay, so introduction. Living things share 8 characteristics. Okay, all living things share these 8 characteristics. The first one is called organization, and that means each organism has a certain way that it's put together that sets it apart from non-living things. It's going to have certain structures. Okay, we know plants have a cell wall. Living things don't have a cell wall, they have a cell membrane. Okay, good. So that's one of the things with organization. If a cell, a tissue, an organ, a living thing moves out of this organizational situation and moves towards disorganization, basically a breakdown in that, that's basically some sort of disease. Okay, that's essentially kind of a funky definition of what a disease is doing. It's taking it from the organized to the disorganized. Responsiveness. Okay, so living things have the ability to respond. There's 2 parts to this. There's what's called irritability, which is an organism responding to change in their environment. So when there's some sort of stimulus, the organism can respond to change. That's its irritability. Okay? The second part is its adaptability. These are the different ways that it could respond to that stimulus. Okay? So on our thing it says the potential options the organism has to make this change. So the different ways it might respond. Okay? When it does respond, that's its irritability. Okay? Any questions on that? Kind of simple so far. If as we go along, I go a little too fast, you need to slow down or something, just raise your hand and say, ho, and then kind of give me some signal that you're ready. All right, till we figure out who's the slowpoke. Then I'll just look at them every time. All right, so third, growth. This one you probably would have gotten. All right. Obviously, this is the ability for living things to get larger. They do this by increasing the number of cells or the size of those cells. When, let's just say a tissue gets bigger because it gets more and more cells, that's what we call hyperplasia. Okay? When a tissue gets bigger because the certain number of cells that they have get larger, that's what we call hypertrophy. Okay? So hyperplasia and hypertrophy are 2 ways that a tissue or an organ, whatnot, could get bigger. Okay? So we could make this class bigger by, instead of having 48 students, we could make it 60. Okay? Or we could lock you in here, feed you nothing but ho-hos and Twinkies, no exercise, and the class would get bigger over time that way. Okay? Which is not a bad way to go, I guess. Differentiation is taking an immature cell and letting it become specialized. All right? So the ability of cells to become specialized. It's a lot like what's happening with you folks. You are all students, so you could all kind of say you're cells that don't have specializations yet. Some of you do. Some of you have gone back to school. But for the most part, you all kind of come out of high school, you have the same educational background. You've been taught basically the same things, maybe a little better, a little worse, different classes, whatever. And now you're going to start to learn specialized things that are going to help you be a nurse or a dental hygienist or a doctor or whatever. So you're going to have different skill sets. And that's kind of what happens to these cells. When you hear stem cells are generic cells. Okay? They're identical, but they have the ability to differentiate into some other specialized cell, a neuron, a muscle cell, that type of thing. Okay? Reproduction, living things have the ability to reproduce. If they don't do that, they don't have offspring. Got to have offspring to carry on the gene lines, carry on the species. Living things have some sort of movement. Movement's kind of in two parts. Some things we know don't really move in their environment, but when we're talking about movement, it can be movement throughout its internal environment. So it moves wastes here to get rid of them. It takes nutrients there to get rid of them. Okay? And then some things can obviously move within the environment themselves. Okay? Any questions on those so far? Got two more left. All right. So next one is metabolism. Okay? Metabolism is a generic term for all the reactions that are going on inside the cell, tissue, body type of thing. So when we talk about your metabolism, it's the rate at which the reactions take place in the cell. All right? There's two types. There's anabolism and catabolism. Anabolism are reactions where they take small units, like amino acids, put them together to make proteins. Okay? They're building. Anabolic steroids build things. They build muscle out of amino acids. Catabolism is the opposite. It takes big things like fats, breaks them down to make glucose for energy. Okay? So you got building up reactions and breakdown reactions. They both fall under the category of metabolism. Now generally, here I'll put the thing up there, chemical reactions taking place inside the body. Now, think about you building something, all right? It'd probably only go to the men in here because the women make us men do that stuff. But like they buy some crazy thing from like IKEA and they say, hey, can you put this thing together? You're a man, you should be able to do that. And they, they don't know. We don't want to do that. And we usually intentionally botch it so we don't have to do it again. But you look at the directions and you work on this thing, and at the end, what do you usually have left over? Extra pieces. And your wife, girlfriend, mom, whoever says, why didn't you use those? And you absolutely have no effing idea why you didn't use those. You really don't. But you say, those are just leftover waste things. We're going to throw them away. Well, anytime you have this metabolism, and they're doing these reactions, there's things, extra parts left over or waste products that build up. So the last thing we got to be able to do is excrete them. Eliminate those things so that they don't build up in our body, because if they do, everything becomes toxic, and that would be bad. We would be moving towards disease. Okay? Who else didn't get the paper? You guys flunked paper passing 101. You just left those people out. Okay. Any questions on that so far? It's not too hard. A little boring, but not too hard. All right. So, regulation. So all living things regulate their internal processes. Okay? And trying to keep those processes stable inside the body is called homeostasis. Okay? So trying to regulate the reactions going on in your body, that metabolism, they say is trying to maintain homeostasis, or a lot of times they'll say trying to maintain a stable internal environment. And when things become unstable, once again, that's disorganization, that's going towards a disease. Now this regulation has both intrinsic and extrinsic factors. Okay? Intrinsic's another word for that's autoregulation. So, let's take a cell or tissue, doesn't matter. Intrinsic factors are things within that cell that measure what's going on and make changes right then and there. Okay? Extrinsic factors, usually from one of the governing systems, another cell that tells that cell what to do. Something maybe your nervous system or your endocrine system tells that cell, we want this or that to happen. Okay, so one's from inside the cell itself and one's from something else. For some of you, your intrinsic motivation for this class is how well you want to do, your own pride in it, how much it matters to you, that type of thing. Your extrinsic motivation might be your parents, might be your whoever's paying for the class, financial aid, that type of thing. An employer that's wanting you to get this so they can move you up in the world. Okay? Intrinsic or auto-regulation happens when the cells or tissues adjust themselves automatically. And then extrinsic is oftentimes from one of the two governing systems, which the governing systems are the nervous system and the endocrine system. Now, a lot of times, this stable internal environment, this organization, things aren't super specific. Did anybody else need this? Are we all good? Cool. Okay. So, there's not a specific value that's supposed to stay in. It's more of a range. So oftentimes we have what are called homeostatic set points. And these set points change or are different already between men and women. Okay? They'll change as you get older. They'll change or be different based on your genetics, what mom and dad passed down to you. They'll change based on your fitness level. Okay? You could take blood pressure, pulse. Okay? There's a range for that that's normal. Not everybody's 110/70. Okay? Their normal might be a little higher, might be a little lower. That might be because they're super fit. Okay? If you're one of those Tour de France triathletes, your blood pressure sitting here in class isn't going to be 110/70. It's going to be a lot lower. Okay? Your pulse rate. Most of us pulse rate average about 70. But once again, it's just an average. You could be normal at 60. You could be a little bit normal towards 80. But if your heart's real efficient, you might have a resting pulse level of 40, 30. Okay? Because it's just that efficient. It's beating that strong with each beat. Okay? Where some of ours are having to beat faster or more often to pump the same amount of blood. Does that make sense? What these set points are? Okay? Temperature even. What do we think of the normal temperature? 98.6, 98.7. Everybody's a little different. That is the norm, but sometimes that's why they even take your temperature when you're not necessarily feeling it. They want to get what your normal is when you're feeling fine. Okay? So due to variability, normal is given as a range, and these ranges are designed statistically to catch about 90% of the population. So that tells us that there's 10% of the population for any given test that will test abnormal while it is still technically normal for them. Okay? All right. Disease. Disease is a breakdown, moving from that organization to disorganization. It's a breakdown in those extrinsic, intrinsic regulation pathways that now we start to move out of that homeostatic range. And when that happens, it's eventually going to lead to damage to that tissue. Maybe if enough tissue is damaged, the organ's not going to function anymore. And oftentimes these are due to an infection, an injury, or bad genetics. Okay? You've got bad kidneys because your mom or dad had them and they passed them on genetically to you. Something like that. Okay? And when we're trying to assess these things, and we're trying to look at these ranges, we have a couple different things. We have what's called a sign. A sign is an objective physical indication of disease. What does it mean when we say objective? Okay, so she's on the right track. It's something you can measure. It's something you can quantify. Okay, this person has a fever because I took their temperature and it was 103.4. Okay, that's a sign. Okay, that makes sense? This person has high blood pressure. Their blood pressure was 165 over 125. I can measure it. Okay, that's slightly different than symptoms. Symptoms are subjective things. They're the things the patient might tell you, but that you can't really quantify. Okay, what's probably the number one symptom that people have when they go to see a doctor? Okay, stomach ache, let's make it even simpler. Pain. Okay, it's the pain. Can you quantify pain? No. Okay, when you come in and you say your stomach hurts, they'll try to. They'll give you that VAS scale. They'll say from 0 to 10 how bad your pain. All right? That type of thing. They're trying to get an idea, but we all know that's different from person to person. Okay? My mother-in-law, she could have a hangnail, and that shit she's going to put 10 out of 10. You know? But you got that uncle, he could fall off a ladder, lose a liter of blood from his head, and you're like, we need to get you to the hospital. Oh, no, I'm pretty good. Just let me sit here for a minute. You know, nothing phases him. It's like an old shoe. Okay? So, you really can't. Everybody's different. So a lot of times when they're doing that, they're kind of measuring where the patient thinks they are and maybe a little bit of their mental state. Okay? If that person writes 0 to 10 and they write 15 over here and they circle it and they start 5 times, they know they're going to probably be dealing with a little bit of a pain in the ass. Okay? So, here's a question, just to see if we understand the last thing there. 45-year-old man presents with pain in his chest and refers down his left arm and shortness of breath. In this scenario, is his pain a sign or a symptom? Still a symptom. Okay, he's describing it. We know what's going on here. This person's, okay, we don't know. This person's probably having a heart attack. Chest pain that refers down their arm oftentimes is a sign of a heart attack or at least angina. Okay, but it's still a symptom. You can't quantify it. Okay, they feel dizzy. Once again, that's a symptom. You can check them to see if they are dizzy, have them do what's called Trendelenburgs and stand with their feet together, look up at the sky, close their eyes, and if they're dizzy, they're going down. So when you do that, you want to make sure you're standing next to them so that you catch them. It's not very professional to watch them fall and be like, oh shit, yeah, they're dizzy. Now, they're bleeding now, but yeah. You have a question? Well, yeah, a symptom you can't quantify, and usually it's going to be something the patient's telling you about what they're feeling, but you can't measure it. Okay, so they could say, I feel hot, or I got the chills. Once again, you can't measure that. You could look for something objective and take their temperature, and that would be a sign. Okay, but it's just basically the objective, quantifiable versus subjective that they're telling you. Okay, when you're charting, or if any of you work in the hospital, I don't know if they have the CNA's chart, but the nurses, if they're doing their SOAP notes or whatever they're doing nowadays, subjective, they're basically what's the patient telling you. Objective, what have they measured? What tests have they done? Have they taken their blood pressure, their pulse? What are those readings? Okay, they put those two together. Okay, so now we start to move a little bit out of the review and a little bit into the anatomy and physiology. And we're going to start off just initially with the term anatomy and what it means. The definition of anatomy means cutting open. Okay? And it's the study of the internal and external structures of the body. Its partner is physiology, how those structures work. Okay? And so this class, we learn a lot of the structures, and we try to learn how they work. Mostly lab is anatomy, learning what structures are what, and the class is the lecture is a little bit more about how do they work, what goes on with them. Okay? Structure equals function. Okay? This is a simple concept. Generally, we might not even know why, but like the appendix kind of falls out of that. Why do we have an appendix? I don't know. Maybe needed it at some point. But generally, cells, tissues, if they have something there, they use it. Okay? So, egg. Okay? Egg from the ovary. Doesn't have to move on its own. Cilia move it to the fallopian tubes, whatnot. So it doesn't have any structures to allow it to move because it doesn't need to move. Sperm needs to move, so it has a tail. The tail allows it to move. Its structure, what it's been given, equates to its function, what it needs to do. Okay? Cytology is the study of cells. And we already mentioned that histology is the study of tissues and how they form organs. And so that leads us a little bit into the hierarchy of things. Organelles are the structures like the mitochondria, the smooth endoplasmic reticulum, all those things, the parts that make up the cell. The cells make up the tissues make up the organs make up organ systems. Organ systems make us up. Okay? So, what's your general definition of a cell? What's that? It's living. Okay. Can we say anything else about it? Yeah, basic unit of all living things. Okay, good. So, cell, smallest or basic unit of living things. Primarily us. Since this is supposed to be human anatomy and physiology, we're mostly concerned with us. Okay. So, cells oftentimes have fluid outside the cell. They'll call it extracellular fluid, sometimes interstitial fluid. There's a slight difference, but we're not going to worry about it. And so it's the fluid that washes around the cell. Some cells, depending on where they're at, have lots of extracellular fluid. Others don't have very much. So that changes from cell to cell, tissue to tissue. What do we call the fluid inside the cell? Well, you could call it the intercellular fluid. That's good. And then somebody, I think, said cytoplasm. Yeah, good. So cytoplasm, and once again, we're splitting hairs here. The cytoplasm is made up of the cytosol, which is actually the fluid. Inside the cell, one of the things that will come into play later on, has a higher concentration of potassium than the extracellular fluid. The extracellular fluid is going to have a higher concentration of sodium. The other component of the cytoplasm, the organelles, are within that cytosol, and then we have protein filaments that make up what's called a cytoskeleton. So all those things combined are what we think of as inside the cytoplasm or making up the cytoplasm. But, like I said, that's splitting hairs a little bit. When we say cytoplasm, most of the time we're really talking about the cytosol, the fluid component inside the cell. And you guys have already told me, but what's the divider between the extracellular fluid and the intracellular fluid or the cytosol? What's that barrier? Cell membrane, okay? So whether we're inside or outside the cell membrane, okay? So cell membrane, sometimes called plasma membrane, separates the cytoplasm from the extracellular fluid. And what is an important characteristic of that cell membrane with respect to letting things in and out? There's a term, 2 words. The membrane is very choosy on what it lets to go. Yeah, there we go. Selectively permeable. Does that sound familiar? The cell membrane is selectively permeable, meaning it controls what leaks out of the cell and what's allowed to come into the cell. Okay? So things can go through the membrane, but the membrane controls that. Okay. Doing all right so far? All right, diffusion. All right, so this is an important concept. It's a simple concept once you got it. Do we know what the heck is diffusion? She's saying combining 2 things. Low to high, combining things from low to high. So together, that's a pretty good answer. Diffusion is moving things from one area to the next, which might be combining, but they're going from a high concentration to a low concentration. Okay? That's what diffusion is. So if we lit a candle up here, one of those really expensive candles my wife wastes money on that smell good, if you lit that, that would start to diffuse throughout the room. Eventually, you guys in the back would smell it. The molecules would bounce off each other, the walls, until it pretty much equally distributed itself throughout the room. Okay? Diffusion. We kind of do diffusion. We get in an elevator, okay? You walk in an elevator, you go like this side. The person behind you, they go to that side. You kind of equally spread out through the elevator. Nobody goes to the elevator and this person goes to the right and you go stand like right next to them. Well, there probably are people like that. They're really weird. We've all had somebody that's just like always in your space. Okay. But most of the time, diffusion, we kind of move, spread things out. Okay? And in this, with the body, we're going to move nutrients, ions, those types of things from diffusion. They're going to come from an area of high concentration, and they're going to move to an area, diffuse to an area of low concentration. Ultimately, evening things out if they can. Sometimes they don't have enough time to totally even things out, but okay, so substance is moving from an area of high concentration to low concentration. Another process which things move back and forth across the membrane is called filtration. Oh, excuse me, I jumped the gun. Osmosis. Osmosis, all came up at once. What is osmosis before it goes back up? There has to do with the movement of water. Okay, good. Now, I'm going to ask before you get up there because I want you to see if you know it. What governs water movement? Does water move from high concentration? Does water move to a high concentration of solutes or to a low concentration of solutes? She's saying water is drawn to salt. Any kind of substance in the water is considered a solute. So she's using salt as our solute. That's fine. So if I had a lot of salt here, not so much salt here, and water could go either way, where's it going to go? Yeah. Water is going to go to the high concentration of salt or solutes to dilute it down, once again, to kind of even it out. Okay? So when you look at osmosis, osmosis is talking just about water moving from an area of low solute concentration to the area of high solute concentration. Okay? So if I had a beaker of water and the same size beaker of water and they're separated by some membrane that let water move, not the solutes. Okay? If there's a little salt here and a lot of salt there, the water is going to go to the lot of salt to water it down. Once again, to kind of even it out. But catch the difference there. Diffusion, the solutes move from the area of high to low. Osmosis, water moves from an area of low to high. Okay? So, once again, it's simple. It's one of those things when you get it, you're like, oh, I got it. That's easy. But I also know I've been there where you're sitting there and you're thinking, what the is he talking about? I have no idea. Okay? Filtration is our last one. Most of us probably understand filtration. Filtration means we're using water pressure to push things across the membrane. Okay? Generally, the membrane is going to be size or shape limiting, meaning the molecule has to be small enough to fit through the holes. Okay? Think of like a strainer that you rinse vegetables off or whatever. You're rinsing that stuff off, or you're rinsing off, or you're putting pasta noodles in there. They're too big. They stay in the strainer. Everything else washes out. Okay? Your pool filter. You push water through the filter. It carries the yuck from the pool that gets caught up in the filter. Okay? Water moves on. Things that are too large get caught up in the filter. Okay? Does that make sense for those three? You feel somewhat okay, knowing that you still have time to study them between the next time I see you? Okay. So, here's a question. We got to put on our thinking caps. So it says, if we have a 500 milliliter hypertonic solution, all right, we don't really care about the size. We just need to know that that solution is called A, and we have another one, 500 milliliters, B, so they're the same size. One's hypertonic, one's hypotonic. So what the foobar does hyper and hypo mean? Hyper's high, hypo's low. So the first one is hypertonic, meaning there's lots of solutes. The second one is low, not as many solutes. Okay? Separated by a membrane permeable only to water. What would we expect to see happen over time? So think about it for a second, and then I'm gonna ask you what you think. All right. So number 1 says volume of solution A would increase. Number 2 says volume of solution B would increase. And number 3, somebody'd shotgun those suckers. All right. Now, how many think it's number 1? How many think it's okay? How many think it's number 2? Okay. And there's probably at least one of you wants to raise your hand for number 3. All right. So we had a lot of you that didn't vote. Kind of like a presidential election around here. Only a few of you vote. Whatever. If you don't care, you shouldn't vote. That's fine. So the answer is volume of solution A would increase. Okay? Water's going to go from the low solute concentration to the high, which was solution A. You might have done this experiment. Okay? It's they put them in little bags. You dump them in the water. One bag will shrivel up. One bag expands and gets bigger. Okay? So when you put the salty solution in regular water, and water can move through that membrane or that bag, it moves in there and causes that one to swell. Okay? Feel getting it a little bit? Okay. All right, so study of tissues. We called that what? Histology, okay? We got about 50 trillion cells in our body making up these tissues. Okay? 200 different cell types. We break these all down and put them together into 4 primary tissue classes. Does anybody know the 4 primary tissue types? There's connective, good. Epithelial. We got 2 out of 4. We got a good solid F right now. We get one more, we go to a C. Nervous. Does that sound right? Okay, so we got epithelial, connective, nervous. There you go. Muscle tissue. All right. Thank you guys for acknowledging that was a muscle. You weren't like blue shirt. Arm sleeve? Makes me feel good. I don't go to the gym to stare at myself for hours without getting good results like that. All right. So, oh, sorry, I didn't put up nervous yet. So, we're going to talk about tissues and these tissue classes and what makes them different and that type of thing. We're going to kind of hit on all of them, but I will tell you that this unit focuses on these 2, epithelial tissue and connective tissue. We'll touch on these, but muscle gets its own unit. Unit 3 is on muscle. Nervous tissue gets units 4 and 5, so we're mostly concerned with epithelial and connective. Okay? So first off, tissue is a group of cells that come together, generally similar cells, and the products of the cells. So the cells produce things that are around the cells that help make it part of the tissue. Okay? Generally, these cells all came or developed from the same region in the embryo. And we don't need to know any more than that. There's different layers. There's endoderm, ectoderm, mesoderm, where all these things develop. But that's more of a developmental biology class than what we need. Okay? And so they develop, they produce things, and those things become tissues. The differences between these tissues are the differences between the cells. So if I have a tissue here, and it has some type of cell, okay, we're not going to worry about it just yet, but we'll call it type A cells. And we have something over here that's made up of type B cells. Would it be safe to say these are 2 different tissues? Right, okay. Now the other things that make up the tissues are what they call cell products, the things the cells make. So maybe we've got similar cells, but maybe these cells are making something different than these cells. That would make them different tissues, right? Okay? And then sometimes the other thing that differentiates them is maybe over here we've got just a few cells and lots of stuff they made. And over here we got lots of cells and they're not making much stuff. But the cells are the same and what they're making is the same, but the ratio is different. That tends to make them different as well. Okay? So differences between tissue classes. Obviously, if the cells are different, the tissues are different. Epithelial tissue is going to have different cells than connective tissue. Oh, yeah. That's more, there's antigens on the blood cell themselves. That's a little different, okay? But your thought process isn't off, okay? Differences in the characteristics of the extracellular matrix. So going back up there to where it said tissue, group of similar cells in the cell products. The cell products are the things that make up what's called the matrix, okay? And they're extracellular, so the things the cells produce that are around the cell. We already know that if there's fluid outside the cell, we're going to call it the extracellular fluid, so that's part of it, okay? All right, so one of those components are called fibrous proteins. Fibrous proteins, fibrous is trying to tell you it's like a fiber. It's very thin and it's long. So it's very thin, long protein filaments, okay, that run in the fluid outside the cell, but help, because this is a tissue, often to give it some sort of shape and structure, okay? Fibrous proteins are solid like the cell. They're not fluid, okay? They're within that extracellular fluid or within that matrix. And then the second component is called ground substance. Ground substance is essentially the extracellular fluid, but it's now in a tissue, so they give it a little different name to set it apart. But it's made up of water, waste products, minerals. Oftentimes it's thicker than water. It's a little bit more of a gel, okay? And it was produced by the cells. So the cells often make up their own matrix. And then the third thing that can make these different that I've already mentioned was the difference in the space taken up by the cells versus the matrix, okay? So once again, it's kind of tough to give you analogies, but if I had like a small fish tank, a small aquarium, one difference between my fish tank and your fish tank is I got half of mine filled with rocks. I've got lots of green whatever stuff and very little fish or water. You've got the same fish, the same rocks, the same green stuff, except you did it right. You put a small layer of rocks at the bottom, some of the green stuff there, and then most of yours is water and fish. We've got two different aquariums, right? Made of the same thing, so it's basically that ratio of rocks to water to fish. This is that ratio of protein fibers to ground substance to the cells. All right, epithelial tissue. So we're going to go into that one first. Epithelial tissue has one or more layers of closely adhering cells. So it has one or more layers, and the cells are packed together tightly. So just from that, if it's telling me that the cells come together tightly, we know there's not much space around the cells for the matrix, right? What makes up the matrix. So epithelial tissue has lots of cells, very little matrix. Generally, these cells form flat sheets. So oftentimes with epithelial tissues, we like to think of them as having two sides. Even though three-dimensionally they have more than that, because they're flat sheets, we think of like a sheet of paper. You see how many sides do you have your sheet of paper? Well, you have two. Okay? There's other sides, but they're so thin they don't count. Okay? Epithelial tissue is avascular. If the cells are so tightly packed together, okay, then it's almost like it's solid. There's no room for blood vessels to pass between the cells. Okay, avascular means without a blood supply. So epithelial tissue tends to be avascular because the cells are so tightly packed together the blood vessels can't get between the cells. Okay? So cells are too close for the vessels. Now, the cells are alive oftentimes and they need nutrients. So the way they get their nutrients, the blood vessels get as close as they can. The nutrients leave the blood vessels that they're in a high concentration in and go to the cells that are using them up, therefore they're at a lower concentration. So how are they getting from point A to point B? If they're in the blood in a high concentration and they're moving upward or laterally or wherever to an area where the nutrients are in a low concentration around the cells, diffusion, there we go. So oxygen and nutrients diffuse up from the underlying connective tissue, because we're going to see one of the differences between connective tissue and epithelial tissue is connective tissue has the ability to be very vascular. So it has a good blood supply. So the nutrients come that far, let them out of the blood, and they diffuse the rest of the way. Okay. So mostly cells, not a whole lot of ground substance, and even fibrous proteins with epithelial tissue. Functions of epithelial tissue. One is to control permeability. Okay? So what does that mean? Permeability. Controls. Do we have anything that was selectively permeable? What did that mean? Chose what moved. Yeah, what came in and out of the cell membrane. So we're going to have epithelial tissue lining the outside of our body for one, and lots of cavities inside our body to control what moves in and out of the cavity, what moves in and out of our body. Okay? So our skin, the outer portion of that's an epithelial tissue. Okay? So when I go get in fresh water, that water doesn't seep into my body and I swell up like a grape. Okay? That'd be bad. Or if all the water when I went out in the sun here when it's 108 degrees left my body really quickly and I'd shrivel up and die like a weed. That'd be bad too. All right? So it controls what goes in and out. So any substance entering or leaving the body must cross an epithelial tissue or epithelium. So controlling permeability. They also, epithelial tissues, provide physical protection. Sometimes these epithelial sheets are in more than one layer. So it's one layer stacked on 100. Okay? And it's on the outside of our body. And if I fall down and I scrape my skin, some of those cells just get scraped off. But guess what? There's a whole bunch of them right underneath there that just take its place. Not a big deal. Okay? So physical protection, abrasion resistance, and obviously preventing dehydration, part of that controlling permeability. So those are some of the functions of the epithelial tissue. Epithelial tissue also produces secretion. Some of these cells have the ability to produce substances and secrete them. Okay? A lot of them line what we're going to learn to be called glands. And this glandular cells coming together we can call glandular epithelium, and they will produce sorts of secretions that we'll talk about. But a simple one that you all know is sweat. Okay? That's epithelial tissue producing a water-like substance that we secrete to the surface of the skin to cool our body temperature. Okay. All right, so epithelial tissue, naming these epithelial tissues, they have two sides. Instead of saying top and bottom, they give them a little bit different names. They call one surface the apical surface. This is generally the upper surface or the surface that is exposed to the environment or a body cavity, a lumen. Okay? So think of your stomach. Okay? Or even your mouth. All right? The outer layer of the epithelial tissue in your mouth opens up into the cavity itself. Okay, that's the apical surface. Deeper, that you can't get to without cutting things out, would be the second layer, which is called the basal surface. So think bottom. The basal surface of epithelial tissue sits on a basement membrane. Okay, this basement membrane is a thin layer of protein called collagen and adhesive proteins that tell you just exactly what they do. They're like adhesive. So the basement or the basal surface of the epithelial tissue sits on this basement membrane. The basement membrane's job is to glue it, essentially, to the underlying connective tissue below it. Okay? So once again, our skin, the outer portion of our skin is epithelial tissue. Underneath it's connective tissue. There's a basement membrane between that anchor them together. Okay? And if you provide enough friction on it, you can actually separate the epithelium from the underlying connective tissue. And it swells up and gets fluid in there. What do we call that? Happens on your feet. Blisters. So a blister is enough friction that you're actually pulling that basement membrane apart, and the epithelial tissue separates from the connective tissue and fills with fluid, and that pressure in there, that hurts. Okay? So naming epithelial tissue, first thing you do is you look at how many layers are there. Okay. And then what is the shape of the apical layer? So the way this goes is if you look at it and you see one layer, you call that tissue simple. Okay? If you see more than one layer, you call that tissue stratified. It doesn't matter if it's 2 layers or 202. It's just now it's stratified. So they're simple and they're stratified based on the number of layers. And then you look at the shape of the apical layer of cells. So we have to keep in mind which is the apical layer. If there's only one layer, that's easy. Just whatever cells we see. All right? And the types of cells that we can have what are called squamous. Squamous are flattened, smashed cells. They look like a fried egg. Okay? You can have cuboidal, that look cube-shaped, square, dice-shaped. Okay? You can have columnar, which look long and rectangular. Okay? There's a couple different options. So when we look at these, and we see this tissue right here, if I was going to try to name that, what would be the first part of its name? Simple. And then we could try to figure out, is that supposed to be square dice-shaped? Is that simple cuboidal, or is that simple columnar? Well, we get the picture. We're looking at the shape now. Down here, what would this be? Stratified. And then we look at the apical layer. Let's say this is columnar. Let's say that's cuboidal. What type is this? Stratified cuboidal, because this is the apical layer. Okay? So it doesn't matter what any of these layers were. All right? And that's how you name them. It's not that hard. Sometimes it's hard because pictures aren't quite that simple, but the concept isn't hard. So we've got simple epithelium, contains one layer of cells. You name it for the shape of the cells that you see. You have stratified epithelium, contains more than one layer, and you name it for the apical cells that you see. Okay. So we're going to try to name a few things here. Probably won't be too hard. So first up, we're looking at this picture right here. These are our cells. What would your best guess to name that be? Yeah. First of all, the answer's right there. So just being a little more awake and you'd all nailed this one. Simple squamous, one layer of flattened cells. Okay? So single row of flat cells allows rapid diffusion of substances. So this is kind of important. When we talked about epithelium, we said we can do protection and we can control permeability. Okay? Now control permeability means, we want to let things in and out, but we want to control what goes in and out. So, if I want things to be able to go in and out, do I want big, fat columnar cells that things have to move all the way through? Or do I want flattened smash cells? Flattened smash cells, good. And once it goes through one cell membrane, and the membrane said it's good, you're good to go, would I need to go through another cell membrane? No. So generally when I'm controlling permeability, I want simple squamous epithelium. Okay? So that when it does let something in and out, it gets in and out rapidly. Okay? Does that make sense? Simple squamous epithelium does have the ability to secrete serous fluid. Serous fluid is a fluid that oftentimes reduces friction. Some examples of where we find simple squamous epithelium: alveoli of the lungs, glomeruli of the kidney, endothelium in the blood vessels. Mesothelium are the membranes that line body cavities inside of us. And these are just allowing things in and out. Okay? And we're going to have this flattened, smashed epithelium. So it's kind of like having a guard gate. If we're trying to get in somewhere, all we need is that one gate. Walk up, they either let us in or they don't. And once they let us in, we're in. We don't need to walk another mile and a half to get to wherever we're going. Just let us in. Or there's no sense going through this gate and then hitting a gate 6 feet past there where they're checking you again. Right? So that's why we want simple squamous. Okay? Now, if we're going to do abrasion resistance, would it make sense to do one layer, simple, or stratified many layers? Many layers. So it's often going to be stratified. It's going to be stratified squamous as well. So those are where you find those two. Okay. So here is a simple cuboidal epithelium. These are supposed to be more square, dice-shaped, one layer. Okay. Then they're spaced. Here's another layer, but they're not on top of each other because this is a duct. So cuboidal, columnar, they're epithelium that secretes things. They make something and they secrete it, and when the body uses it somewhere else. Okay? So simple cuboidal is to usually produce some sort of secretion. It's not concerned about protection. It's not concerned about abrasion resistance. Okay? Single row of cube-shaped cells, and the function is absorption and secretion. Now, in our body, the things that usually produce secretions are called glands. So you tend to find these in the mammary gland, salivary glands, thyroid gland, liver, which isn't a gland but does produce secretions, or any other gland, is where you're going to find simple cuboidal epithelium, because that's where we're producing the secretions. Okay. Now, we might list that once in a while you might find a stratified cuboidal, but it's very rare, very uncommon, because think again, once again, about the structure and the function. If this cell's secreting something, okay, that's its job. It wants to secrete it, put it into this duct. Does it do us any good to put another cuboidal cell right in front of it blocking it? No. Okay? So it's not that it can't happen. It's just not the normal way you're going to find it. So really, you're going to find lots of simple squamous epithelium. You're going to find lots of simple cuboidal, simple columnar. When you find stratified, it's almost always stratified squamous. Okay. So, simple columnar. Here, these are long rectangular columns of cells. Here's the basal portion. Here's the apical portion. Single row of tall, narrow cells. Because they're so thin, the rounded nuclei get smashed and they make them oval-shaped. Absorption and secretion, once again, is their function just like cuboidal. And here we actually have some goblet cells, or what they're called. They make mucus. And you can find these lining the inner lining of the GI tract, the uterus, or the kidney. So oftentimes simple columnar will have these goblet cells associated with them, or they're just going to, for whatever reason, they're going to produce mucus. Okay? Mucus is sticky. All right? It absorbs water oftentimes and becomes sticky. So this you're going to find doing kind of a protective thing, not abrasion resistance, but secreting this mucus to the apical surface so that if anything from the outside world tries to make its way through it, it gets caught up in it like flypaper. Okay? So we want simple columnar epithelium, usually lining anything that the cavity or the pathway is exposed to the outside world. That's why you find it in the GI tract, because food from the outside world comes into our body that way. Uterus, reproductive tract, the JJ, we want it there. We want to catch any wee beasties that might be trying to make their way in there. Urethra, males, we want to catch it for wee beasties crawling in your junk. Catch them there in the kidneys. We don't want our kidneys to get infected. Okay, so once again, they put them, or the body puts them there because they have a specific job. Okay, it doesn't do any good to do their job inside a blood vessel because we're not going to find stuff there. We're going to keep stuff out before it even gets to that point. Okay, so here's one that I didn't mention because it doesn't fit the rules. It's kind of that outlier, and it's called pseudostratified ciliated epithelium. And pseudo means not stratified. So what happens with pseudostratified, here it looks kind of like, because when they stain it, the nuclei are easily seen. It looks like we've got one layer here and another layer of these purple nuclei here. But what's happened is not all these layers have grown all the way up to the top. So the ones that have, their nuclei sit about midway up. And the ones that don't forces their nuclei down. So it gives this kind of looks stratified appearance. So they named it pseudostratified, not stratified, even though it looks that way. Okay? Now, it does essentially the same thing as simple columnar. Okay? So single row of cells, not all of which reach the apical surface. Uneven appearance of nuclei gives it this stratified look. Now, this pseudostratified always tends to have cilia. Cilia are like little hairs. And these little hairs beat and they move. So here, when they produce the mucus, things get caught up in it, and the cilia move it one direction or the other to wherever they want to get rid of it. Okay? And you tend to find these pseudostratified ciliate epithelium in the respiratory system. Okay? So that when you breathe in the air and you get something bad in there, hopefully it's going to get caught up in the mucus. The cilia move and actually beat it upwards towards your mouth, where you swallow it back down into your stomach and your stomach acid kills it. Okay? People that smoke a lot, the first thing that happens is it kills those cilia. Okay? They lose the cilia don't move. So these people catch stuff in their throat or the respiratory system, but they can't just move it up there and swallow it. How do they get it out? Anybody that's coughed or smoked for years, they have a what? Smokers cough. That. They're hacking it up into their throat to swallow it back down. And you smoke long enough, this will actually change from pseudostratified to, I think, stratified squamous. You stop smoking, over time it'll kind of come back, but it's kind of like gaining weight and losing weight. I can put 10 pounds on in a week. It'll take me 6 to lose it. Okay? So found in the respiratory system. All right. That takes us to our stratified. So those were all the ones that were basically under the simple, even though the last one didn't say simple. It just says pseudostratified. It still was one layer. Stratified epithelium has more than one layer of cells. We named them for the apical layer or the basal layer. Apical, good. Name for the shape of the cells on the top or the surface, aka the apical layer. We're going to have another exception to the rule like we did pseudostratified. This is going to be called transitional epithelium, but we'll get to that. Okay. The deepest cells, the basal layer, still sits on top of a basement membrane. The basement membrane anchors it to the underlying connective tissue. It also restricts proteins from diffusing upward to the epithelial tissue. and it essentially glues it or fixes it to the underlying connective tissue. Stratified epithelium can have a couple of variations. We can have what's called keratinized epithelium. Keratinized epithelium, the layer, the apical layer of cells are dead. Okay? Non-keratinized epithelium, the layer of apical cells is alive. It lacks this keratin, so it lacks the dead cells. Okay? So, inside your cheek, you have non-keratinized epithelium. Outer layer of your skin, you have keratinized epithelium. This layer of cells on the top is dead. Okay? This apical layer of cells is alive. Anybody had to look at a cell under a microscope in a bio class? Where'd they have you swab it to get it? Cheek. Okay? They had you do the inside of the cheek. They didn't say scrape it off your arm because it's a dead cell. You won't see anything. Okay? These dead cells slough off all the time. Okay? But they're dead. You can't see anything. You have to get one that's alive. Okay? Keratinization kind of waterproofs the skin. It reduces or slows down the water loss out and the water coming in. Okay? So that's why we have it on the outside of our body. But on the inside, they're still alive. There's no keratin, so the surface layer of cells is alive. So here's a picture of keratinized stratified squamous. Here's the basal layer that I'm tracing. Okay? It's not even, but as we can see, we just had to know, is there more than one layer? Yes, absolutely. Okay? Right in here, we start to produce the keratin, and you see the cell shape goes from at least having some thickness to having no thickness. Because when we put the keratin in here, these cells all got nutrients from the connective tissue below. It diffused up. Okay? Went from a high concentration to a low concentration. But that keratin not only prevents the water loss, it prevents the nutrients from moving through there. So now these cells get cut off from their nutrients and they die. Okay? And so this is keratinized stratified squamous is good for abrasion resistance. Okay? Outside of our skin. So multi-layered, surface has dead squamous cells packed with the protein keratin. The keratin decreases water movement, does a really good job of it. It also is going to prevent microorganisms, wee beasties, pathogens, or whatever from making their way inside the skin. And the main place that we find this keratinized stratified squamous epithelium, I've been referring to the skin, is called the outer layer of the skin, which is known as the epidermis. Your skin has an epidermis and a dermis. The outer layer is the epidermis, and it's stratified squamous keratinized epithelium. Try to kill one more slide here. Not the last one, but we have keratinized, so we have non-keratinized squamous, stratified squamous. And so once again, here we have the cells, the basal layer. We don't care what the basal cells look like. They look cuboidal. Out here, these are flattened and smashed, so they're squamous. Stratified squamous. We don't have the keratin. It's got the more than one layer. No surface cells are dead. This is also good for abrasion resistance, but we want it moist and slippery. So the skin, we don't need to be slippery. Inside your mouth, you want it to be slippery. You want the food to slide back. You want it easy to swallow. When your mouth's dry, it's kind of hard to swallow, isn't it? Places that you find non-keratinized stratified squamous. Tongue, oral mucosa, esophagus, vagina. Okay? So those are all places that the surface cells are still alive because there's no keratin. All right, we'll stop it there for today. Be prepared that we could have a quiz on Tuesday. See some of you in lab. Otherwise, have a good weekend.